All right, welcome everyone. Thank you so much for joining. This is the monthly iMapInvasives webinar series. This is our March 30th webinar, and today our topic is 2021 user stories. So we're very lucky to have some great speakers here with us today, and I'll list out their names. So there's Garrett Chisholm from the EN Huyck Preserve. There's Dr. Christy Tyler from the Rochester Institute of Technology. We have Abigail Bezrutczyk and Haley Gladitsch from the Long Island Invasive Species Management Area. And we have Emily Caboot from the Averill Park Central School District. And so I'll start by giving a couple highlights from NY, from NY iMapInvasives from 2021. But most of this webinar is going to be focused on hearing from some of our super users who I just listed out. So to start with my brief highlights, so for anyone who's not familiar, iMapInvasives is it's an invasive species database, an online database and mapping tool used by several states and provinces across North America. In New York, it's the centralized database to support PRISM, state agencies, and other partners working on invasive species issues. You can view distributions, generate reports, set up early detection alerts, and more. And to give you a snapshot of the data that came in last year, you can see over 20,000 records, presence records entered into iMapInvasives, and then we also had a bunch of not detected in treatment records as well. And I also wanted to acknowledge that we are part of this larger network, so it's not just New York reporting to iMapInvasives. There are several states and provinces across North America as well. And we generated an annual report together this year. So if anyone wants to learn more about that, you can visit the website listed here. And I'll send out a link including that link as well. And you can see, so there were over 20,000 from New York for presence records, but you can see there's even more when you add in all the records coming in from states like Pennsylvania and Maine, all the way up to Saskatchewan. And on the report, we also list out some of the new updates to iMapInvasives from the last year, like, for example, more options for filtering data. And there's also each participating jurisdiction, we actually have a couple more now, but at the time when we created the report, we each shared a highlight or story. And for New York State, we shared our spotted lanternfly mapping effort that we coordinated with the New York State Department of Ag and Markets. And just to give a brief progress report of how that went last year, we were very excited to have a lot of participation, a lot of data submitted across the state by our volunteers. So if anyone's not familiar with this, the idea is that there's a sign-up map, and people select a location to survey for spotted lanternfly and tree of heaven. And these locations were grid squares that you pick. And so we actually had 108 grid squares with both tree of heaven and spotted lanternfly data in them by the end of 2021. So hundreds of records of spotted lanternfly and tree of heaven, and we're really excited about that, and we're launching it again for 2022. So you can feel free to check that out at nyimapinvasives.org. And then my last slide before, or actually two more slides before switching it over to our panel of users, just a couple more quick highlights from 2021 from the NY iMapInvasives perspective. So we did the 6th annual mapping challenge in July, got hundreds of records of some key underreported species across the state, and got to hand out fun trophies to our partners at the PRISMs across the state. We got the invasive species tier list table up online so that people can look at how species are ranked in different regions of New York, and you'll hear more about that later, with how the Long Island Invasive Species Management Area has been using their tier list. And we've had more and more users this year really getting into the idea of standardized reporting of presence and treatment records to iMapInvasives that allow for comparisons over time. And we're really trying to get that moving for water chestnut in particular. And so in this example, you can see water chestnut was reported in 2020 and then reported in 2021. And we have used the information in those records to estimate biomass. And at this particular site, we can actually demonstrate that the biomass has decreased probably in response to treatment. So we're always excited to see that sort of story in the data. Most of all, we wanted to thank everyone. So everyone out there who uses iMapInvasives, give yourself a pat on the back. We really value your contributions, and we hope that the database is continuing to be useful for all of our partners. And with that, now we can shift over to hearing from the users. And so our first presenters will be Abby and Haley from the Long Island Invasive Species Management Area. Take it away, Abby and Haley.
Thanks, Mitch. Yeah, I'm Abby Bezrutczyk with the Long Island Invasive Species Management Area, and I'm joined by Haley Gladitsch. And LIISMA is one of 8 PRISMs in New York, or Partnerships for Regional Invasive Species Management, but we use iMap and its resources all the time in all areas of our work, really. All the work that we do that's listed there. So being able to have data on hand about an invasive species allows us to coordinate our work in management, surveying, monitoring, and connecting partners. To know where species are is just essential. And we can use this iMap data to share information with our partners, bring the local community together, and raising awareness about invasive species. And lastly, as we'll talk more about, iMap helps us assess and rank invasive species so we can actually do the work that we need to do. Next slide. I mentioned that LIISMA is one of 8 PRISMs, which you can see on the map there. We have a pretty unique area across Long Island, Brooklyn, Queens, and Staten Island. For starters, the whole western end of the area is highly urban to suburban environment, which means there's a lot of people and a lot of places that have been exposed to invasive species, places where they've been able to take hold for quite a long time. But on the other end, out east, we have a high quality rare species communities through pine barrens and even coastal plain ponds, as pictured in that image, with a high diversity, so worthwhile areas to protect as priority areas. So having such a diversity of areas with sometimes extreme proliferation of invasive species means that prioritizing our work is a very important way to operate. Next slide. So this is where the tier list comes in. Not only do we rank species according to their impact level and distribution across our PRISM, as you can see in that chart right there, we now, thanks to NYNHP and former grad student Dylan Finley, had the opportunity to see how these rankings compare across PRISMs. So at LIISMA, we gave it the nickname the Big Tier List. And the Big Tier List is really essential for us. For starters, to prioritize our work. So since we have such diverse ecosystems and populations, and a great proliferation of invasive species, a lot of species in this case we categorize as tier 4. And there you can see, I counted out, we have 96 species that we have in tier 4, which means local control. And to some that might be disappointing because it means that, for example, knotweed, a tier 4 species, we don't control in all areas, like if it's in somebody's backyard. We don't have the staff capacity or resources to respond to that. But it goes across scale. So we might manage knotweed in areas that are high priority water bodies or terrestrial areas. So having this tier list allows us to focus on emerging species before they establish here as well, and while providing also a framework to clarify and standardize our decision making to the public. And while the big tier list doesn't have regulatory power like county do not sell this or the Part 575 regulations, it's still really actionable in guiding where we work and what we pay attention to, in making recommendations to partners, and also horizon scanning, so seeing what species are behaving invasively elsewhere so that we can be on guard. And it takes us also, lastly, from a reactive approach to a proactive approach. While it's not always perfect, just like human decision makers aren't always perfect, it's a step in the right direction that really helps our work, and we use it all the time. So I'll pass it off to Haley to talk a little bit more about it.
Yeah, thanks, Abby. So yeah, diving deeper into the tier list. The tier list really helps us to have more efficient conversations with different prisms and to really understand how prisms near and far rank different species. And this helps us to see either if there's an emerging invasive species that we should be keeping an eye open for, or understand if there are inconsistencies with how we might be prioritizing different species. Siebold's viburnum, for example, is considered a tier 3 or containment in the lower Hudson, but tier 5 or a research species in LIISMA, the one that we're trying to really understand its distribution and abundance in our prism. That makes me think that a conversation about this species would be useful in how we approach categorizing it or managing it. Should it maybe have a lower ranking, move from a 5 to a 3 because of its growth habit, or should it remain a 5 because of its moderate ecological impact? And while I don't currently have the answer to this question, just having the language to ask it makes a world of a difference in working towards that answer. It really helps us to continuously perfect how we think about ranking these species individually and across genera. It's not perfect, but it's extremely functional. Another example is Tree of Heaven, which is considered a tier 4 in LIISMA and our closest neighboring regions, but it's a tier 2 in, and 3 in APIP and SLELO, respectively. Which means in LIISMA, we aren't actively removing every tree that we come across or that we hear about, while folks in these more northern prisms are and have a lot of experience doing so. And this has been extremely helpful recently as we've been considering working with some of our partners on Long Island to consider managing some of these Tree of Heavens in priority areas with the emergence of spotted lanternfly. And again, having that language to have these conversations makes them so much more efficient, because then I was really easily able to head to the New York Natural Heritage Program page and hear or find out about what a species rank is, Tree of Heaven in this case, and then automatically know the management goals of these other prisms, and then have a better idea of who to specifically ask advice about maybe some of their management goals and what they've had successes and failures on. Go to the next slide. So the tier list within our prism, just going even deeper into it, it's really helpful in thinking about how we manage invasive species across the state, as I've described, but as we look more finely at the priorities in our region as well. Statewide, Phragmites is a tier 4 species, meaning that pretty much everywhere state agencies don't have Phragmites eradication programs. Meanwhile, with giant hogweed in the state, it's a tier 3, meaning there's a goal of containment all across the state. And there are programs dedicated to it within New York State. But then when we look more finely at the prism level, Phragmites is still a tier 4. So within LIISMA, again, we're not going everywhere trying to remove Phragmites personally, and we're not setting aside a lot of time and energy to do so. On the other hand, on the prism level, giant hogweed is now a tier 2 species, meaning within our region, our goal is eradication. There isn't a lot of it. We can hopefully try to eradicate it. And we work with the New York State different agencies to do so. And then even more finely, at the site-based level, again, giant hogweed, still a tier 2, removing it wherever it occurs, whether it's in someone's backyard or a high priority conservation area. But Phragmites, on the other hand, at the site level, this is where that local control really comes in these high priority areas. Whether it's an ISPZ or a heritage site, we can really start to think about, okay, if we see the species there, we're gonna remove it. And so if we see it in an ISPZ or in a heritage site, like I said, if we wanted to make a management plan, we could then try to develop a tier list specifically for that site. So Phragmites would then, on that site-specific tier list, we would call that a tier 2 there, because that site might have the goal for eradication in the example I gave at Sears Bellows. But again, at this site, there isn't a lot of barberry. So on their tier list, barberry might actually be a tier 1, even though all across LIISMA, it's considered a tier 4, it's widespread, it's everywhere. And again, so this language can really help us to more finely prioritize, not only on the state level and within the LIISMA region, but on that really fine grained level with our partners to help them figure out what their management goals really are. You can go to the next slide. So yeah, you can see when we did find Phragmites at Sears Bellows, we knew to stop everything because high priority site, there isn't a lot of Phragmites there. We knew to eradicate it from the site, get the permits going, which is what we're doing now. You can go to the next slide. But yeah, so we really love the tier list. It's not only been super useful in talking to other prisms and land managers, but in having discussions with the general public. it really helps us to back up and why we prioritize what we do, and goes a long way in helping folks to understand what is in our capacity to do, and learning about the widespread versus emerging invasive species within our region. And then from there, how to really interact with LIISMA when they do come into contact with either. In fact, I think my favorite moment with the tier list was outside of work when I wasn't clocked in. I was in a class at the New York Botanical Garden talking about fall perennials with a bunch of horticultural specialists, and Chinese millet grass came up, and I was able to pull out the tier list on my phone and show them that this is a species that we consider a widespread invasive, even though it's not necessarily regulated or prohibited. And it was really cool when a lot of those hort professionals were able to say, "Oh, can you send me that link? Can you email that to me later?" And it really goes to show this is a tool that can be used not just for us and the conservation-minded folks, or people within the state, within these agencies, but really horticultural folks and gardeners, people that might see these species at a gardening store, or aquarium people who might be buying aquatic plants online that might be considered invasive, but might not yet be regulated or prohibited, as well as anglers buying bait, etc. But yeah, so I'll leave you with this. When Abby and I started, we kind of got started as the tier list was coming onto the scene. And so even though it isn't perfect, it is a living document that changes as we learn about the species. We really, at this point, we just couldn't imagine doing our jobs without it. So yeah, big thanks to everyone who put in all the work to make it as great as it is.
Thank you so much, Abby and Haley. That was great. It was really great to hear how you're using the tier list, communicating with partners and as well as the public, and thinking about it differently at different scales. That's really awesome. Thank you for sharing that. If anyone has any questions for Abby and Haley, please feel free to enter them into the chat, and we should have plenty of time at the end to go over any of those questions. So thank you again to Abby and Haley for speaking on this. And now I will give it over to Garrett Chisholm with the Huyck Preserve.
Thank you, Mitch. My name is Garrett Chisholm, as Mitch said, and I'm the Stewardship Coordinator for the Edmund Niles Huyck Preserve and Biological Research Station located in Rensselaerville, New York. My primary responsibilities as Stewardship Coordinator are to help oversee our land management operations. So a little bit about the Huyck Preserve. We are a nonprofit LTA accredited land trust with over 90 years of continuous forest research, and our mission is divided into 4 parts to support conservation, education, research, and recreation. So our preserve has over 2,000 acres of land with 12 miles of trails, and our property consists of 71 different ecological communities, 15 of which are rare or imperiled at the state level. So our relationship with iMapInvasives began in 2019 when we drafted our invasive species management and monitoring plans that was later adopted by our board in 2020. This is all thanks to funding provided by the Capital Region PRISM through their RFP program. Next slide, please. So iMapInvasives is utilized in 2 different facets of the preserve's operations. The first being for monitoring and managing Hemlock woolly Adelgid, which, as many of us know, is an invasive aphid-like insect from Japan that feeds on hemlock trees and was first discovered here in 2015. The photo on the left I took of a recent infestation I discovered. The second is being through monitoring and managing our terrestrial aquatic invasive plants, which we have upwards of 30 different plants that we manage here at the preserve. Next slide, please. So as I mentioned, iMapInvasives is used heavily here for Hemlock woolly Adelgid monitoring. At the preserve, we have over 350 acres of hemlock stands, and based on various criteria from ecological significance to cultural value, we have established 6 different priority stands, which you can see here on the map of the preserve. So over the winter, I spend a great deal of time monitoring our majestic hemlocks here at the preserves. The only tools I bring with me are a super strong flashlight and the iMapInvasives mobile app. So iMap has really made monitoring our hemlocks here quite seamless. And with my eyes being the only ones out in the field monitoring and IDing for these hemlocks, it's been really great to have my observations confirmed by various professionals through iMap, and has even helped me better understand, where my own misidentifications could have occurred. So monitoring here between 2020 and 2021, which is what the map is displaying, revealed that we had Hemlock woolly adelgid infestations in 5 of our 6 priority stands, with each of these points on the map being from the records that I had uploaded to iMapInvasives and since transitioned over to ArcGIS Pro. So as I had said, we had only had by 2021 Hemlock woolly adelgid present in 5 of these stands. However, monitoring this past year in 2020-22 has revealed that the infestation has now spread to all 6 of our priority stands. So this map will be looking much different once I finish up my monitoring this year. So though a lot of this is negative with Hemlock woolly adelgid, there have been some positives as our data that we've uploaded to iMapInvasives has helped support the need for this management, and we've actually been able to treat 2 of our 6 Hemlock stands thanks to successful applications again through the Capital Region PRISM's RFP program. So we treated stand number 3 in 2020, and we treated stand number 1 in 2021. Treatment being a basal bark spray consisting of diamethyfuran applied to the base of the trees, and imidacloprid, which provides a long-lasting protection and prevents the spread to uninfested trees. An injection of imidacloprid was actually used when trees were closer to the water. And again, thanks to PRISM, we will be able to continue this treatment in 2022. Next slide, please. So iMap is used heavily, as I mentioned, for data collection here for our terrestrial and aquatic invasive species, and it's used in almost all aspects of our management. So in the summer, I hired 2 assistant invasive species workers, and so we have a crew of the 3 of us. And so when in the field, myself and the invasives crew will log all presences using the iMap mobile app before any mechanical removal will take place. So after the day's project has been completed, whether that be moving a small patch of pale swallowwort from deep in the woods or chipping away at our large population of Eurasian water milfoil via kayak from the lake, we sit down in the office and review what we logged that day on iMap and then copy those records into our own internal database before uploading. So you can see on the left there, I used an example of a false spirea plant, and you can see our log in the top right corner. And then after all of our records have been uploaded, we then go in using iMap's online interface to log all of our treatments, which example of you can see in the bottom right-hand corner, again, for false spirea. Next slide, please. So one of the most helpful aspects that I found personally in my work from iMapInvasives is being able to regularly download and utilize all of the data stored in iMap. So because of all of our data being uploaded to iMap since 2019, we have a significant number of invasive species points to work with. So I'm able to export all of our data from iMap and upload it onto ArcGIS Pro. And so once I've uploaded it to ArcGIS Pro, I then go through and separate the data into individual shape files for each species, which is displayed with a different color point. So we're looking at ArcGIS field maps, and on the left is the outline of the preserve with each and every one of our invasive species points, which can look quite overwhelming. But once that data has been sorted, I create this map on ArcGIS Online, which can be displayed through field maps or another ArcGIS app such as Collector or Explorer. And this has been immensely beneficial when planning and doing the actual on-the-ground field work, as you can turn on and off various layers and navigate on the ground to the specific infestation that we would be tracking that day. So if you can see from these screen grabs, I turned off all layers except for autumn olive. And you can see on the right side of the screen where the autumn olive populations are specifically here at the preserve. And this has been really great for, days that I'm not able to go out with my crew. I can give them instructions in the morning, and they have all the information that they will need via this app. And since all of the data from iMap is attached to each individual point, we're actually able to see what was logged in the past while being live out in the field. Which has been really great if some of the points are tricky to find or we need information on past management practices. Next slide, please. So I wanted to end out on 2 of the more exciting highlights from our work at the Huyck Preserve this past year, and that is some of our experimental projects that we have done with invasive species management. So first, we used a portion of our 2021 Capital Region PRISM RFP to request a weed torch to use on select more widespread species here at the preserve. So we set out to manage plants in areas of high visibility and ecological significance along the trails that surround our Lake Myosotis and Lincoln Pond. And so during our stewardship intern week, during what I called an invasive blitz, we went out and hit Japanese barberry, common barberry, multiflora rose, and autumn olive, cutting them back with loppers. And then after about a month, I went back and sprayed each individual plant with water from a Smith Indian fire pump, and then briefly blasted them with the flames from the weed torch, making sure to not completely burn the plant. So after about a month, I went back and monitored and could not identify any further resprouts from those plants. And so since this experiment occurred later in the growing season, additional monitoring will be needed in early spring to help finalize and see our results. And that is a photo of myself on the left using the weed torch on a patch of multiflora rose. So the hardware cloth experiment was recommended to us by Chris Williams at Capital Region Prism. And again, with our Prism RFP, we purchased the hardware cloth to try experimental management on Japanese knotweed. So we have two populations of Japanese knotweed that are adjacent to Lake Myosotis. And this project was developed to test the effectiveness on the management of Japanese knotweed. And what we hope to see is the knotweed being girdled as the experiment will take place. And so this patch was managed. We have two patches, one being managed normally and one being managed with the hardware cloth. And so all the above ground biomass was removed from both patches, and the hardware cloth was laid out on one of the patches to be labeled as the experimental plot, while the other was left as a control. So I then went in and created four 1 meter by 1 meter study plots marked with red flagging, as would have been shown in some photos after on the right. And then after about 30 days, we returned to the plot to count the number of stems and measure the height of each stem per plot. So the initial results of this experiment did show that there were more stems in the experimental plot, but those stems were shorter than that compared to the control plot. And notably, neither of these populations, when managed, went into flowering that season. More information, again, will be needed to be gathered through future monitoring, but we think that this may be an initial response to the stress caused by the hardware cloth. And both of these treatments, to bring it on back to iMapInvasives, were logged. The weed torch treatment being logged under flame weed in the treatment area, and the hardware cloth being logged under barrier. And that is the end of my presentation there. I'd like to say a quick thank you again to Mitch and iMapInvasives for hosting us for this webinar today. Thanks.
Yeah, thank you so much. That was a great presentation, Garrett. It's great to hear iMap being used at this local level, reporting treatments, as well as follow-up surveys to assess whether the treatment's successful. And yeah, so thank you so much for presenting that. And one thing this makes me think of is how there's many different ways that one can go about recording their surveys and their treatments in iMapInvasives. So Garrett's kind of worked out this process, which works well for the Huyck Preserve, where they report invasive species while they're out in the field, and they do their management. And then when they get back to the office, they go in and enter those treatment records. And that works well for a lot of groups. Some groups prefer alternatively to actually record those treatment records while they're out in the field, using either our Survey 123 form or our iMap Mobile Advanced, which is in field maps, Esri field maps. So there are different ways to go about it, and it kind of depends on which process works best for you. So if there's anyone out there who's trying to figure out how they want to collect treatment data in iMapInvasives this field season, please feel free to reach out to us, and we're happy to talk to you about figuring out the process that works best for you. And I'll share my email at the end of the presentation. So thank you so much, Garrett. And
thank you.
Yeah, thank you. And I'll now pass it on to Dr. Christy Tyler from the Rochester Institute of Technology.
Okay, hi everybody, and thanks Mitch for asking me to share a little bit of what we're doing. Mitch asked me to talk a little bit about how we're using iMap in both teaching and research at RIT. I'm Christy Tyler, and I'm a professor in the environmental science program here. And so I'll give a couple examples of projects we've been working on. Next slide. All right, so the first one I wanted to talk about is our senior capstone course. And this is a course that's required of all of our environmental science majors. And students work in teams with a community partner for the full year. And one thing we started 8 or 9 years ago is when we start a new project, we want it to run for a minimum of 5 years. So all of our projects are long-term and there's kind of continuity among years or across years, which has helped us to be able to accomplish bigger projects. And all of the projects incorporate some kind of data collection that ends up with a large database, different types of deliverables and reports, and then all students do outreach. Next slide. So the project that's been most involved with the iMap group is a project that's kind of morphed into the overall goal of improving community science engagement with invasive species management programs. We've, over the last, it's been, I think, almost 9 years now, we've worked with iMap and also the Finger Lakes PRISM, the Federation of Lake Associations, and then also have interacted with the Kinesis Lake Association. Next slide. So the first phase of this project started back in 2013, and the students that worked on that were most involved with the NICE-FOLA, so the lake associations, and they worked on merging the iMap data with the Citizen Statewide Lake Assessment Program data, and created these map books for different lakes in the state. And they did over 100 lakes where they created these map pages that are shown on the right. The students became trained in iMapInvasives and then became certified trainers. So they conducted a number of different trainings, both on campus with student clubs and courses, and then also off campus. So for example, the picture in the bottom is the students doing a training for the Kinesis Lake Association. And the overall goal of this was to create more awareness of aquatic invasive species. But one thing we discovered as a part of this, and they had done some community surveys with the Lake Association, is that the iMap records underreported what we knew were out in the lakes, in spite of the community being really aware of invasive species and of the problem of invasive species. Next slide. So then the next phase of the project, we moved more towards trying to understand barriers to community engagement. And so that started in 2018. We started to work with Hilary Mosier and the Finger Lakes PRISM. And in particular, we're using the Macrophyte Survey Program as a case study. The students conducted both qualitative and quantitative studies. They did interviews. They've conducted a number of different participant surveys, both with iMap users and for the Macrophyte survey participants. Based on those results and literature review, they've created engagement plans. Using the Macrophyte survey program data through iMap, they've created story maps. They have also interfaced with our Google Street View project, which I'll talk about in a second. They continue to become certified trainers, each team does, and then they've also been able to present in multiple public forums, including at the International Association of Great Lakes Research Conference. So overall, it's been a really successful project, and we have a group who's working on it right now. Okay, next slide. Okay, so then this is what I alluded to, this is a research project where we've been able to involve some coursework, but also some pretty innovative research. The first bullet is noting my colleagues on this. Chris Cannon is in our imaging science department, and he's a computer vision specialist. And then Arturo and Manoj and Liam are students that have been working on this, and I've highlighted Liam because most of the slides and the figures that I'm going to show he made. So he's been really instrumental as the liaison between the development of the model and the iMap team. So the premise of this is that Google has collected more than 16 million kilometers worth of this roadside panoramic imagery. So the photograph on the right shows a knotweed infestation on the side of the road. So by using computer vision, there's a lot of promise for being able to detect roadside plants without as much effort of going out and actually monitoring. So this project's now funded by the DEC, and our goal is to create model output for the 5 species at the bottom. So Phragmites, knotweed, purple loosestrife, parsnip, and tree of heaven. Next slide. Quick overview on the model mechanics. We took initial data from iMap and found infestations that were close to roadways and used this as the initial kind of training data for the model. And from that did an iterative process of training the model with additional photos, testing the model until we have, for the first 2 species were Phragmites and knotweed, close to, at this point, we have close to 7,000 panoramas that have been viewed. The model creates a heat map, which is what's shown on the right, where it believes one of the invasive plants are and calculates a probability that it's right. And then you get the model output from that. Next slide. So this is just showing how we incorporated the project into a course. This is a freshman course, and the students were trained in iMap by the senior Capstone students, so they knew how to go out and monitor species. And the original plan with this is that the students would aid in the field validation for the project, where we would take the model prediction and actually go out and check. But then COVID happened. This course was being taught in spring of 2020. So we revamped the plan and had the students do an on-screen assessment comparing iMap data with the model output data. So in the map on the right, that's Schuyler County. This is the Phragmites predictions. Are the colored dots, and then the little black diamonds are where the iMapInvasives records are. So you can see that there's a concentration of the iMap records in Watkins Glen, but there's Phragmites that hasn't been reported throughout the county on the major roads. So the students in the class then conducted some literature review, and I had them write a weed management plan as their final deliverable for the project. Next slide. So when we came to doing the actual model validation, which COVID put off a little while, we generated model predictions in 11 different areas and did first an on-screen validation and then did a field validation, and that was done through iMap using an iMap user group in order to validate the predictions from the model. And this is just some of the statistics from that, but the true positive plus true negative rate, so basically when it gets it right, is somewhere between 50 to 90%, depending on the species and the location. But more importantly, the false negative rate, so the number of times that it misses a plant being present, is very small. When it came to the, so that's doing the on-screen assessment. So and then the field assessment, you get a true positive rate of greater than 90%. So if the model says it's there and you look on the computer, say, yes, I agree it's there, chances are really good that it is there unless it gets mowed down, which is the case in a number of the different validation locations. Next slide. So one of the pieces that Liam's been working on, this is just the proof of concept for this, but it's to compare the model prediction with what records already exist in iMapInvasives. And there's a huge amount of new data that comes in as a result of this. So this is a conservative estimate that he backed down based on how often the model actually is correct. But it's adding thousands of new records beyond what was already present in iMap. So next slide. And then Liam's also developed this public interface, and this is the idea of being able to have community scientists volunteer to do the model validation, either on screen or actually going out in the field. So this is the dashboard that he's developed. And these are the points that have already been validated. But the goal will be to have this be able to go live so that once the model development phase is done, someone can go through and move them from kind of these leads and needs of observation type of records to a confirmed record based on an actual evaluation of the image. Next slide. Okay, and then it would show this is just some of the, whether it's been confirmed or not detected based on the data that's there. So that is my last slide. Thank you. I'm happy to answer questions. Oh, no, it's not my, I forgot about this one. I can't forget about these guys. Okay. I wanted to give a quick example of something because I know the next presenter is going to be talking about K-12 and iMapInvasives. But these are, this is my daughter's Girl Scout troop, and we were able to have them complete the community science journey for seniors and then also their silver award, which they did on invasive species. And so they used iMap in a local nature area to map out where the invasive plants were. So they made these maps. They chopped down as much autumn olive as they possibly could. And they got a lot of recognition for this from the town. So it was a really successful project. And having them able to use the app and see their records online was really validating for them. So it was a really great tool for them to see the efforts that they were putting in. Okay, that's my last slide. Thank you.
Thanks so much. That's awesome. So many different projects that your group is working on and using iMap in so many different ways. And yeah, if anyone has any questions, please feel free to enter those into the chat box. And so thank you so much to Dr. Tyler for her presentation. And that brings us to our last user story for today, which comes from Emily Caboot with the April Park Central School District. She'll be talking about how she has integrated the invasive species curriculum into her classroom. And a fun fact about Emily is that she led the creation of this, working with DEC and other partners. She led the creation of the curriculum for her master's project a few years ago. So now I'll hand it over to Emily.
Awesome, thank you. My name is Emily Caboot. As Mitchell said, I am the creator with the help of many people in a work group from many years of work during my master's time to create the curriculum. A little bit about myself now. I teach high school in April Park, and if you look down in the corner, that is, it shows you where it is in New York State, so it's in Rensselaer County, and then the town in the county is red as well. We're a rural school district in the mountains of Rensselaer, and so I teach living environment, which is biology, and then horticulture as well for the high school. This year, as is my first year actually in the high school, I was in the middle school previously, I was able to implement my invasive species curriculum, and I decided to do it in the ecology unit for our living environment class. And I also did it in my did portions of it in my horticulture class. As we maintained our garden area, we have a large garden with a greenhouse in an area where we compost. So I was able to introduce invasive species to many of them and actually maintain some areas. So for the class that I focused mostly on doing the curriculum was my living environment by my biology students. Those are 10th grade students. And most of them knew of invasive species, but I was able to really help them dive in and identify many that are in the area. It was very easy to implement with my students, and they also really enjoyed it. They loved going outside, and at the beginning of the school year, especially this year, which was our first year with all the students back due to COVID, they really enjoyed going outside, taking walks, identifying things. And at the end, which I'll talk about briefly afterwards, we were able to go out and start to maintain some of the areas, and we actually got to eradicate some of the invasive species from our bus loop. All right, next slide. So just a little bit of what I did with the curriculum, because I didn't do it straight through as I originally designed. I worked it into the end of my ecology unit. So in the beginning of the unit, we just focused on the basics of ecology, how things cycle. But then I weaned this into the last 2 weeks of the unit as like a capstone kind of project to it, and worked in biodiversity and other topics. So that's where it's plant identification for the students, because in the living environment curriculum, there's not a ton of plant ID or really any kind of ID of any living organisms, surprisingly. So in the curriculum, which is online, it's planned out, but this is just a snapshot from the beginning of the curriculum. We went through what invasive species were and how they spread. So the students did research. I helped them through to develop a focus or an end product, like a scientific poster that college students usually create. And we walked through creating an introduction, identifying plants, creating methods, going outside and completing field work, and then using that information or using the data that they collected to create some sort of visual to make sense of what we found. And then we actually went ahead and did a conclusion writing and then got to get outside and eradicate some of the invasive species. Next slide. So this is one of the examples. I let the kids try to get a little bit of creativity with this. Students worked in groups of 3 to 4, depending on who they wanted to work with. And we walked through, which on the left is the introduction for what invasive species are. So I had a little bit of lecture time with the students discussing, having some phenomenon. And then I allowed the students to research through a collection of sources that I gave them to figure out what invasive species were. Then I led them through most of the methods of what we were going to do and had them go back and tweak if they changed. And then we used iMapInvasives, so I did a little training with them to be able to use that app out in the field and collect data. There's images here of the invasive species that they found. We focused on having each group only uploading 1 point to iMapInvasives, so we don't have an extra or a abundance of the same information in the system. And then they went ahead and concluded their invasive species research. And at the end of this entire unit, the students shared their information because a lot of it was similar with the invasive species introduction, but what they found, the different invasive species they found, were, there was about 8 that we found on campus. Next slide, please. This is just another example from 2 of my students who work together. Again, introduction is on the left. We went through, they took many pictures. I had a lot of fun, interesting images with the students that I just didn't want to share because their faces were in the images. But they got outside, especially some of my energetic afternoon students really had a fun time finding the invasive species, taking images, identifying them, and then mapping them. At the end, we were able to go outside after we took our ecology test, and we spent about an hour going to the locations that they found the invasive species and started eradicating them. And then I showed them how we get rid of them. And that's why I brought my horticulture students in to help them dry out and kill the invasive species before we toss them away. Just as this was my first year doing this in my actual classroom, we weren't able to go on a field trip due to COVID, but next year I'm hoping to be a little bit more in depth with some of the mapping portions of the iMapInvasives because the students really were interested in that, and then also take them to a local area. So I think that is my last slide. So I'll take any questions. And also actually my presenters prior, I learned a lot of stuff that I'd like to integrate into next year. I know the Huyck Preserve, you're not too far from us, so maybe we'll come visit and see your fire experiments because the students absolutely love the fire management. Absolutely love it.
Awesome. Thank you so much, Emily. It's great to see the curriculum implemented. And so I'll thank all of our wonderful presenters. Thank you so much for taking the time to share your experiences from last year. And also in the participants list, I see a bunch more of our super users. So I'd like to thank all of you as well. So thank you to our PRISM partners, to our state agency partners, particularly with the Department of Environmental Conservation, the Department of Ag and Markets, and the Office of Parks, Recreation, and Historical Preservation, and then our partners from other organizations as well, like the Huyck Preserve. So thank you so much to all of our iMap users and to everyone who participated today.